All right, so now what happens during the stages of mitosis and meiosis? So why are there two phases? Why is there mitosis and meiosis? They both start at the same point. DNA has already been replicated, and the cell has grown large enough to divide. So, the cell might look something kind of like this. It's elongated, but the DNA is all kind of distributed throughout the cell. And so it's ready to divide, but you have to actually now segregate the DNA in a proper way. And so the way you segregate the DNA is what's different between mitosis and meiosis. And so the goal of mitosis is to make new cells with two exact DNA copies of each chromosome. So you want all of the cells to be exactly the same as each other. And this is used during development. So you start with a fertilized egg, and then eventually, you have the replicated, to trillions of cells. So if I look in my skin cells, if I look in my neurons, if I look in my heart cells, they all have the exact same set of content of DNA. And this makes sense. You don't want to have each DNA cell to be its own unique copy, some from your mother, some from your father, or something like that. You want all of it to be a diploid cell that is exactly the same. And so that's an important thing to remember. The goal of mitosis is to make two new cells with exactly the same DNA. Meiosis is used during in the germline in order to produce gametes. And so one of the things to remember is that gametes are haploid, and so you actually need to half the original DNA content in order to create a haploid cell. And the other thing you want to do is you want to make sure that all of your gametes are very different from each other. So, you might have siblings, and so you should know your siblings are very different from each other unless you have an identical twin, and then you might know that your identical twin is very similar and much more similar to you than kind of a brother or a sister. And so that is because each gamete has a unique content of DNA within it, and so that makes sure that the offspring are all different from each other. And that's one of the goals of meiosis, is to do that. So we're going to talk about mitosis first. So again, this is the whole cell cycle, and then the textbook, the Klug, actually defines mitosis into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. And this is a little bit unique. Many textbooks don't actually include prometaphase, and I'm not going to cover this in much detail, so you don't really even need to know the details of prometaphase. So the important ones that I want you to remember are prophase, and I think of that as preparation phase. This is when the cell starts to prepare to separate the DNA. Metaphase, I think of this as midplane phase. This is when the chromosomes go to the midplane. Anaphase, this is the active phase. This is when actually the chromosomes are separated. And then telophase is the termination phase. This is when cytokinesis occurs and the cell actually forms two new cells. So understanding the stages of meiosis. So your textbook has these nice pictures. So these are actually real life pictures of a cell using stains, and then schematics that kind of show you what's going on. So interphase, this is not a phase of mitosis. Remember, interphase is all of the cell cycle except for mitosis or meiosis. But it has this nice schematic that shows you how the DNA looks, and so you can see that the DNA is pretty much completely disorganized within the nucleus. Prophase, this is when this condensation occurs, and so you go from this disorganized organization to this condensed and coiled up, where you start to see what looks like chromosome structures. Prometaphase, the chromosomes are clearly double structures, and the centrioles reach the opposite poles, and the spindle fibers form. So the centriole is what is actually pulling on the chromosomes in order to separate them, and this acts as an organization point for all of these microtubules. And so at this point, you actually start to see the centromeres form so that you see this very clear double structure. So I'm kind of lumping these two phases together in terms of what happens during prophase, like many textbooks do. So these chromosomes then need to align at the midplane, so the midpoint of the cell, and so this is kind of equidistance between the two centrioles. And at this point you see all of these chromosomes are moving and then kind of orientating and rotating, and so that they form this nice line of chromosomes. So you can see this is chromosome 1 right here, and then this is a homologous chromosome 1. And so we often use color in order to distinguish between the paternal and maternal contributions. So this could be the father's chromosome 1 that you inherited, and this is the mother's. And then these are both chromosomes 2, the father and the mother's. But you can see that the homologous chromosomes are kind of randomly spaced apart from one another during mitosis. Anaphase, so again, when we go back to the schematic of what a chromosome is, it's DNA molecules that are held together by these proteins. And then the goal of anaphase is to actually separate these chromatids from each other by breaking apart these protein bridges. And so once that occurs, you can actually pull this chromatid to one side and this chromatid to the other side. And so that's what the schematics is actually showing you, is that you've pulled apart these chromosomes into two chromatids, and now these are moving to separate parts of the cell, either end of the cell. And then finally telophase. Telophase occurs, the nucleus reforms, so this DNA is compacted into separate nucleuses. And then cytokinesis starts, where you actually start to see a pinching here, and the cell is actually going to divide into two. So at the end of telophase, you would actually have two different cells. All right, so let's skip to meiosis. So meiosis in a lot of ways is very similar to mitosis, but there are some really important differences that you have to remember. So one thing that's very important is that there's actually 2 rounds of meiosis, and that is why you go from a diploid cell into a haploid cell, because there's these 2 rounds of meiosis. And so that first round is called Meiosis I, and so that results in the formation of 2 different cells. And then after that you have Meiosis II, and then those 2 further divide, so you actually form 4 cells. So one of the differences between mitosis and meiosis is that mitosis forms 2 cells, meiosis ends up forming 4 different cells. These are both diploid. Mitosis forms diploid cells, and then the meiosis forms haploid cells because of this extra round of division. Another important thing that occurs is that you have recombination occurring, and so this is actually between homologous chromosomes. And so here we see these homologous chromosomes are pairing, and they're actually breaking and exchanging parts of their DNA molecules with each other in order to form these new chimeric chromosomes that hadn't been seen before. And so again, just remember, in mitosis, you never have any sort of pairing going on between the homologous chromosomes, but that is not true during meiosis. In the first round of division of Meiosis I, you're actually separating out the homologous chromosomes. So this structure right here is the tetrad, and so it's named kind of for obvious reasons. You see the 4 different chromatids that are all lined up together, and so this tetrad actually forms, and it actually separates so that each cell gets its own unique version of the homologous chromosomes. And then after that, Meiosis II, this kind of more matches mitosis, where you then further separate out the sister chromatids and break apart those protein bridges in order to separate the chromatids into single cells. So let's go through the processes of meiosis in more detail. So prophase I is the first stage that we're going to go over in more detail. And so again, they both have these multiple prophase, metaphase, anaphase, and telophase are also in meiosis. And then we name whether or not they occur in the first round or the second round by using this 1 or this 2 afterwards. So prophase I is the first stage of meiosis. And so this is such an important phase, and this is such an intensely studied phase by researchers that they've actually further divided prophase I into these 5 different substages. I am not going to test you. You do not need to memorize leptonema. I don't even know how to pronounce them, to be honest. Zygonema, pachyonema, diplonema, and diakinesis. But what you really need to understand is kind of the process of what the cells are going through during this prophase I. And so, again, prophase preparation, you have this initial organization. So in this stage right here, you start to see that organization really occur, where you're now seeing this compaction occur pretty much, so you can start to see things underneath a microscope, for example. But what is really important is that what you're seeing here is actually a pairing between the homologous chromosomes. So again, this doesn't happen during mitosis. This only occurs during meiosis I, and specifically prophase I, where these homologous chromosomes are actually finding each other. And so you see what's called a bivalent. And so this bivalent occurs when you can see the 2 homologous chromosomes pairing up with each other, but the organization hasn't reached the point where you actually see the centromeres pinch in so that each chromosome has that kind of characteristic structure. So just as a reminder, kind of during prometaphase, we saw this change into this. And kind of the way they're depicting this here is a little bit different. This is actually kind of two sister chromatids. So this is 2 DNA molecules within this single blue line, which can be a little bit confusing. So eventually this organization reaches the point where you see what's called a tetrad. And so this is just because this chromosome has organized to the point where you can see the 2 sister chromatids separately. So they start to see space in between these. And you can also see the nucleus is still there. It hasn't broken down quite yet. The nucleus doesn't actually break down until the end. At this point, in the 4th stage of prophase I, you have this really interesting thing occur where there's actually genetic exchange between them. And so what this means is that you're creating new combinations of alleles and new combinations of DNA that hasn't been seen before. And so what you give on to your offspring is not what you receive from your parents. So again, you receive a chromosome 1 from your mother, and a chromosome 2 or chromosome 1 from your father, but what you end up giving to your offspring is a chimera that is a little bit of your mother's and a little bit of your father's. And so this is part of the reason that your offspring or your siblings don't look like you, is because you not only randomize the chromosomes, but you also randomly splice together chromosomes together in order to make this mix and match. And so this actually occurs during prophase I. So you see that this kind of exchange is occurring here and here, and as a result, you actually break DNA molecules and ligate them to the different DNA molecules in order to do that. And then the final stage is when this process completes during diakinesis. And then the centrioles move to the ends of the cell. So these are actually those kind of showing where the microtubule organizations centers are and kind of moving to the right place during this prophase I. All right, so metaphase I, now we have these chimeric tetrads, so the homologous chromosomes are actually moving to the midplane, and it's important to remember that actually what gets separated during anaphase I is that you're separating each of these chromosomes, these homologous chromosomes, to two ends of the cells. And so again, these are kind of chimeric, so that's where you see this kind of orange and blue combination comes from, is because you've made these new combinations. And then telophase I is where you create the two new cells, very similar to mitosis. Prophase II, nothing really interesting goes on. And then metaphase II, you now line up the chromosomes at the midplane, and the purpose of this, again, is to break up the sister chromatids from each other. So this is when the sister chromatids are actually broken apart from each other. Those protein bridges are destroyed, and then you move a chromatid to each of the cells. And so in the end, you see you only have one copy of chromosome 1 and then one copy of chromosome 2. And so this is why we say that meiosis, so one of the things we say meiosis is that it's non-conservative, and that's because you'll notice that each of the cells has a different complement of DNA because of this process. So if you look at the color, that kind of tells you that. And so this is a very random process. So part of the randomization is where exactly these breaks occur, and then part of the randomization is the pairing between the different chromosomes. And so every time you do this meiosis, that's unique, and so that's why each individual is so different from each other, is because of this kind of meiosis process. All right, so why does meiosis but not mitosis generate variation? So one reason is recombination. So this creates variation within the chromosome. So this is where exactly those chimeric fusions occur. And then the other reason is that homologous chromosomes are randomly segregated at anaphase I. So this part right here. So in this particular thing, this chromosome went here and this chromosome went here. So you had a primarily blue chromosome matched with a primarily orange, but that was just a random process. And so it could have been the blue and the blue could have been paired together and gone to the same cell. And so this also creates variation as the combination of alleles between the different chromosomes as well. And then just finally, we will talk about some of the further details. So pairing and segregation of sex chromosomes during meiosis. So sex chromosomes, so if you are a male individual, male sex, you typically have an X and a Y chromosome. And in that case, you don't have two exact homologous chromosomes. But during meiosis during meiosis, these X and Y come together to form their own version of a tetrad. So the X and the Y actually pair together. They have regions that are homologous, which the cell uses in order to actually match the X and the Y together. There is a minor amount of recombination in what's called these pseudo-autosomal regions, but these are only regions that are very similar to each other on X and Y. So in general, there's very little recombination that occurs, and that's important because the gene content on X and Y is very different. So Y chromosome, for example, has genes that actually specify the male sex and contain proteins that actually confer the male sex onto individuals. And then during anaphase I, these separate apart. So again, you break up the homologous chromosomes during anaphase I. And then just some of the specific details that are going to be important for the third part of this lecture is we are also going to go over a little bit of how actually gametes are formed in mammals. So these are male and female precursors. So these have undergone DNA replication, so they've replicated DNA, and now they're preparing to go through meiosis. So the male version of these cells are called spermatogonium, and the female version of these cells are called oogonium. Sperm development leads to the development of these spermatozoa, and this basically follows kind of the process of meiosis that we went over in the previous couple of slides. So you end up forming 4 independent gametes that have their own unique combination of DNA, and then these differentiate into these spermatozoa cells. Female development is different because you actually only end up forming a single cell as a result of meiosis. So you form 4 spermatozoa as a result of meiosis in the male, and then in the female you only form 1 ovum. And so the reason is that one of the cells actually undergoes apoptosis. So you can see you've had this first round of meiosis, and you have this oocyte, and this second cell actually forms what's called a polar body, and then this polar body actually goes on and undergoes apoptosis and never actually enters meiosis II. And then during meiosis II, you create 2 cells, but the second one is this smaller polar body, and this again does not undergo differentiation, but actually undergoes apoptosis. So you only actually form 1 ovum. And it's thought that this is involved in quality control. This just ensures that the ovum that is formed is hopefully a very healthy ovum that did not have any sort of kind of errors occur. And then the regions that had errors occur, the parts of the cell that might have had errors occur, are hopefully segregated into the polar body. All right, and then this just kind of little table that you can look at your leisure kind of helps sum up all the differences between mitosis and meiosis.